I wanted to thank Dr. Lisa Cunningham from the NIH for an incredibly elegant, beautiful talk. I mean, really, truly from bench to bedside, how basic science can inform translational research, which then can inform our clinical research. Dr. Cunningham, thank you very much. And Dr. Herzana, tell us about the ANS Research Grant recipient.
Thank you. So, as y'all know, the American Neurotology Society offers 3 grants a year, and I'd like to introduce Dr. Dunia Abdul-Aziz from Harvard, that is the 2019 ANS Research Grant recipient for her project named Targeting Epigenetic Modifying Enzymes for Hair Cell Regeneration. Dr. Abdul-Aziz obtained her Bachelor's of Science and MD degrees at Harvard, where she also continued as an otolaryngology resident, neurotology fellow, and in 2018 as a surgeon scientist faculty. Dr. Abdul-Aziz has been working in collaboration with Dr. Albert Edge on the role of epigenetics, which in other words is changes to the overall packing or structure of DNA in directing hair cell fate. She has functioned as an active surgeon scientist from as early as her residency, I actually remember meeting her in the lab. Her pioneering work extends from organoids, which are many engineered ear-like structures in a dish, to in vivo studies in mice, and uses cutting-edge genome editing techniques such as CRISPR-Cas and viral gene delivery to unlock the path for hair cell regeneration. Dunia, congratulations for the award, and we're all looking forward to your very exciting talk.
Good afternoon, and thank you for the American Neurotology Society for funding this research and for allowing me to present it today. I have no conflicts to disclose. The prevalence of sensorineural hearing loss and the general irreversibility of this loss once it occurs motivates our efforts in the hair cell regeneration field. Ultimately, the crux of the problem is that hair cells are lost, and in humans, they're largely cannot be restored, such that cumulative injury from noise, drugs, and aging result in progressive deafness. However, based on developmental programs in mice, as well as our understanding of lower vertebrates such as birds, we know that hair cells can be restored. In addition, we take motivation from other organ systems such as the intestine, where regeneration naturally occurs. So how do non-mammals regenerate their hair cells? Briefly speaking, non-mammals regenerate through recruitment of surrounding supporting cells, which following damage and loss of hair cells either go on to divide or differentiate to restore the hair cells. And among cells that are mammals and non-mammals that do and do not regenerate their hair cells, the basic machinery is largely conserved. And one key gene involved in this is called 801, depicted here. And while driving 801 expression is able to restore hair cell regeneration during development, it ceases to be the case with age. And the question is why that is. And so we and others have hypothesized that epigenetics may play a role. So epigenetics largely refers to how DNA is compacted within the gene. Depicted here on the left are histones, which are tightly packed, making a gene in this area inaccessible for transcription. And so the idea is that there are several chromatin marks which mark whether a histone is tightly packed or not, and that over time course of aging and maturation, there's an accumulation of repressive marks and a decrease in activating marks. And indeed, one such gene which seems to have manifest this pattern is 801 itself. Dr. Noah Siegel's group has looked into chromatin changes that occur during the development in mice and found that during late embryonic stage as well as early postnatal stage in mice, there's a decrease in activating marks and an accumulation of inhibiting marks. And motivated by this, we used an organoid system and a drug screen for epigenetic modifiers in the lab and identified one possible epigenetic modifier known as lysine demethylase 1, LSD1, which may play a role in this process. So my work is ultimately to try to better understand this. So one thing about LSD1 is that its function in the ear is largely unknown, but it's in canonical form in other systems. It acts as a repressor. An interesting thing, however, is that an additional variant of LSD1, which is a splice isoform, which incorporates exon 8A, which is a 3 amino acid loop in the activating domain of LSD1 converts it from a repressor to an activator. What's more is that it's known to be uniquely expressed in neurons and promotes neuronal differentiation and neuronal maturation. So we wondered whether these different isoforms may be playing a similar function in the ear. We started to study this role of LSD1 in the ear using an organoid model. In this model, we can take newborn mice and dissociate the supporting cells and grow them in culture, allowing us to expand supporting cells over a 10-day period and subsequently induce differentiation to hair cells. And what's remarkable about this process is that they really do regenerate hair cell-like structures, which express stereocilia. And we can then go on to study such things as hair cell development, genetics, and perform drug screens. In addition to treating these cells with drugs and studying their function, what I developed is a viral-based approach to genetically alter these cells and study mechanism. At outset, I sought to investigate the effects of LSD1 on hair cell differentiation. So using organoids generated from the 801 NGFP mouse, in which GFP reports on 801 activation, I was then able to study the effects of LSD1 inhibition over time. And what we found is that addition of an LSD1 inhibitor augmented the standard differentiation cocktail, which is using Wnt activation and Notch inhibition in a dose-dependent fashion. We then went on to study the expression changes that occur during this process and found that indeed using LSD1 inhibitor, we could potentiate expression of key hair cell genes such as 801 and GFI1. What is the role of the LSD1 8A isoform in the ear? At onset, we first looked to see whether the 8A isoform was expressed in the cochlea and confirmed that to be the case. So in the early postnatal as well as adult mouse, the 8A isoform was expressed. And then we looked to see whether it was also expressed in our organoid model and found that indeed it was. And intriguingly, we found that the LSD1 8A isoform expression came on with differentiation. We saw no expression of the 8A isoform during the progenitor stage, but then progressive increased expression during differentiation. What's further, I took this and looked at specifically whether the 8A isoform was expressed only in hair cells and found this to be true. Using the organoid system to generate hair cells, which are again marked by GFP, we can then sort these hair cells for GFP and assay for expression of the 8A isoform specifically. And depicted here is qPCR data showing that the GFP positive cells indeed do express characteristic hair cell markers, as well as the 8A isoform in red. We then were interested to figure out what the function of this 8A isoform was. And given its function in neurons, where the 8A isoform seemed to potentiate differentiation and maturation of neurons, we hypothesized that it would perform a similar function in the ear. With this, I delivered targeted short hairpin RNA against the 8A isoform in the organoid model, marked here by red, and then subjected these cells to FACS sorting, and then looked at the proportion of GFP positive or hair cell positive cells among the culture relative to control targeted RNA, and actually found the opposite to be true. Targeting the 8A isoform with targeted knockdown of the 8A isoform actually resulted in increased expression of the 801 GFP. In a complementary experiment, I overexpressed the LSD1 8A isoform during differentiation and assayed its effect. In contrast to the neuronal system, where the 8A isoform was actually sufficient to induce differentiation, we found no such thing to be the case, and no induction of hair cell genes with overexpression of the 8A isoform. Moreover, when I combined it with Wnt activation and Notch inhibition to induce hair cell differentiation, I found that overexpression of the 8A isoform was actually inhibitory to hair cell differentiation. This led me to conclude that both the LSD1 and 8A isoform in its canonical and non-canonical forms function to repress hair cell differentiation. And now in these initial experiments, we were mainly querying a few hair cell genes, and the possibility that the 8A isoform may be functioning more later in maturation remains to be possible. And so we will be studying this using global expression changes with RNA-seq. Our ongoing experiments, which are still inconclusive, is to study whether these organoid findings translate both to induce hair cell regeneration in hair cell damage models, both in vitro using explants as well as in vivo adult system. And these experiments are much more laborious and require a lot of mice. So stay tuned. What I've hoped to have shown you is that the canonical LSD1 as well as the LSD1 8A isoform both inhibit 801 and hair cell differentiation, and that interestingly, the LSD1 splice isoform 8A is uniquely expressed in hair cells. And our ongoing experiments look into global expression changes associated with LSD1 inhibition and LSD1 8A knockdown, as well as validation of our organoid findings in vivo. What I hope to have shown here is a framework for studying hair cell differentiation using targeted viral delivery in our inner ear organoid model, which can then be applied to any other epigenetic modifier or target. And with that, I'd like to acknowledge my advisors, Dr. Albert Edge and Dr. Yang Shi, a previous postdoc in the lab who started the LSD1 work, my research team, and my funding sources. Thank you.
Thank you so much, Dr. Abdul-Aziz. We don't have any questions in the chat box right now, so I think we'll move on to the next session.